Hello, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Leslie Mitchell, who is a pioneer in synthetic biology and genome engineering. She has a PhD in systems biology from the University of Ottawa and a postdoc across both Johns Hopkins and NYU, where she was involved in a pretty groundbreaking project involving creation of a synthetic yeast using genome engineering. And today, Leslie is the CEO and co-founder of Neochromosome, which is a biotech startup that's really taking, I think, a pretty fascinating approach to deliver entire chromosomes designed and built de novo into host cells. And we're going to learn a lot more about this. This is one of our first episodes, I think probably the first episode, going deep into the world of genome engineering and yeast and model organisms. We've done a little model organisms in the past, but not a deep dive like this. So I'm super excited for it. And Leslie, thank you. Really appreciate you taking the time.
Happy to be here.
I'd love to just hear about how you got interested in this field to begin with. What was your first exposure to synthetic biology and genome engineering? And then maybe you could walk through a little bit of your research career in both Ottawa and JHU and NYU that I mentioned.
Yeah, happy to do that. I think it's sort of all wrapped up into one story. And that is to say, I was wrapping up my PhD in Ottawa. It was yeast genetics related work. I had to invite an external examiner to be part of my thesis defense. And so I reached out on a whim to Jeff Buca, who is sort of a world-renowned geneticist. He was at Hopkins at the time, and asked him to join my thesis committee, and he did. And so he showed up and gave a seminar the day before my defense, and I learned about this concept of, rather than studying genetics and biology by breaking things, which is to say change a gene and see what happens, building from scratch. And the concept of the meta question being, do we know enough about biology, genetics, et cetera, to design whole genomes from scratch, and sort of test and extend our knowledge around biology by building de novo entire genomes. And so this to me was like mind-blowing. And I thought, this is something I could get into, all the caveats of breaking things and seeing what happens. Does the antibody have specificity, blah, 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 all of those questions that felt like this was like this totally new approach. And so luckily I gave a pretty good thesis defense. And over the course of that day, Jeff's flight was canceled and he spent the day. So we spent a bunch of time together. And by the end of the day, I had an invite to join his lab. And so that was exciting and certainly career altering. I had not planned to do a postdoc. And so that is how I got off onto this sort of synthetic genomes career track.
Amazing. And maybe you can talk a little bit about what you accomplished during that postdoc, some of the big projects. Sure. And sure. The field underwent some amazing transformation during that time. I remember I was a PhD student at the time, not at all involved in constructing artificial genomes, but watching some of the papers coming out and being amazed actually at, to the point you made earlier, what became possible in that period of time. So maybe you can talk a little bit about that.
Oh, sure. Yeah, the first synthetic cell from the Venture Institute, I think, was in 2010. And so this sort of just preceded my completion of the PhD and sort of joining Jeff's lab. But the big project in Jeff's lab, a little distinct from the original synthetic cell in the Venture Institute, was not to rewrite a whole genome from scratch. And by rewrite, I mean take the sequence that's known in databases and just remake that basically same sequence, but just do it synthetically. The concept for what is now known as the Synthetic Yeast Genome Project, or SC 2.0, was to take the wild type sequence and in silico make designer changes across the length of that genome. Yeast has a 12 million base pair genome length, 16 linear chromosomes, genes, centromeres, telomeres, repeats, the whole nine that everyone's used to when thinking about genomes, but make changes across the genome as part of the design that would allow us to open up new, otherwise unapproachable questions with respect to the downstream biology we could study. Examples, let's delete all the repeats. Are they really necessary for a genome to function? That's a simple one. tRNAs, which are a source of genome instability, let's move those all into one place and take them out of the chromosome positions. Does the cell function? Is the genome more stable? Remove introns. If you remove introns, can then you remove the spliceosome, which is otherwise essential? Are there other essential functions of the spliceosome? all these questions related to how could you ever answer them unless you designed the genome from scratch and eliminated or modified all those features. The final change that's kind of groundbreaking is the ability to, with a chemical switch, change the entire genomic architecture of the designed and sort of synthetic organism. And that is to say, the world's greatest acronym, SCRAMBLE. Let me see if I can get it right. Synthetic chromosome recombination and modification by LOXP-mediated evolution.
Amazing.
I haven't said that one in a while, but peppering the genome with LOXP sites to enable chemically induced recombination globally. Obviously, a lot of cells will die. The deletion of an essential gene would cause death, but every cell that survives, you're going to have essentially in parallel millions of new genomes to search for interesting phenotypes, excuse me. And so the scramble system was, I think, probably one of the features that put everyone over the top. Back in, I think this was like 2007. What do you need to do to justify writing 12 million base pairs of genome? Back then, that was going to be like $1 or $2 a base pair or something like that.
Wow.
And so, it's a big decision to make.
Yes.
How do we even get to the end of this project? But anyway.
Yeah, I mean, you're blowing my mind with some of these fundamental questions. I'd love to actually take some of those in order and just know what the answers were, because I come from the rare disease and non-coding genome world, and I was also in humans wrestling with the question of what is all of this genome doing here, and is it important, and what happens if you delete big chunks of it? And you only see that in humans in rare disease cases or in population genomics programs. But to the point you made earlier, it's subtraction rather than addition, and you don't have the toolkit. So for some of those questions, what did you learn?
Well, we're getting much, much closer to the answer. And I think there's a lot to unpack here. I will say some key points to make here. There's been two major milestones associated with this SC2 project. And of course, in the academic world, that tracks back to publications. Yep. Well, I should say three. The initial publication, which was sort of 2011, there was a batch of papers released in Science in 2017 describing many of these features, how to build chromosomes, how to consolidate them, that they all seem to work and are stable, and then use of the scramble system. And then just about 2 weeks ago, there was another big batch of papers released in Cell. So at this point in time, the status of the project, 100% of the designed DNA is in cells. Wow. That is to say, there's 16 strains in the world, each of which carries one synthetic chromosome.
Wow.
There's a series of strains with increasingly more synthetic chromosomes consolidated. And all of this has been described now. So we're at about 50% consolidated into one cell. So we don't necessarily have the answer to all of these questions yet, which is to say, we don't know about the introns. We don't know about the repeats. All signs point towards no major functional consequence to deleting all of those elements in the context of individual chromosomes, or the most we can say is up to 50% of the cell. So it's kind of a remarkable thing. And I think there's so many learnings from this project on the technology side. How do you design, build, deliver, evaluate function, debug design features that you didn't really anticipate causing fitness defects? But certainly how long it takes to get to the answer. Yes. And we'll talk a little bit about Neo as we go forward, but one of our goals is to start to figure out how to do these sort of things, these types of projects a lot more efficiently.
Yeah, it's exactly where I was going to go with it. What did you learn about how to build chromosomes during that 7 or 8 years of your life that you spent here? Yeah. And how do we take it going forwards? What does it take? What does it cost? How long does it take today? And then what does the future look like?
Yeah. When you kind of look at SC2, the project is probably going to be about 15 years end to end ish, 50 or 60 like core scientists plus an enormous number of additional supporting cast members, including a lot of undergrads who participated in this project over the years. I will note they all show up as authors on these papers, which is very exciting for them. Yep.
Yes.
And millions and millions of dollars. How this all got funded is a separate conversation. But how do you go forward and instead of 15 years, do this in a year or a day? We're definitely not at a day yet, but I think we have line of sight on a year for 12 million base pairs. I think that's really, really the realistic number. State of the art is still order synthetic DNA in small pieces, and how quickly can you assemble it into large pieces and then install that DNA into the destination cell. And are you going to install it as an add-on and then sort of eliminate the original, or are you going to just do like a swap and replace right up front and then sort of do that iteratively depending on the cell type and the sort of length of DNA you can deliver in a single shot? And so there's enormous amount to unpack across all of that. But in short, hierarchically is your answer, the TLDR on that.
Yeah, so maybe you can talk a little bit about the inspiration behind Neochromosome, why you decided to start a company and move out of academia. And then I'd like to get into the mechanics of actually how these things are built. Are they printed? Sure. Are they synthesized? Sure. But first, I'd love to hear about how you decided to start a company.
Sure. Well, my life goal was never to be an academic, and I appreciate a lot of things going on in academia, and there's also things that I struggle with. So I had always wanted to go into industry. That's number one. Number two, we had this sort of big technology stack, at least in principle, which is to say, how do you think about a lot of experience, let's say, thinking about designing whole genomes, the yeast genome in particular. As an aside, I stopped working on ST2 probably around 2017 and turned a lot of my attention to taking this technology and putting it into mammalian cells at the 100 or 150 kb length range. And so how do you design sort of generically large pieces of DNA for many different organisms? How do you build it really, really quickly? And quickly is still on the order of weeks or months. How do you deliver it? And that's now cell type specific. And then what validation steps do you need? Sequencing for sure, but phenotyping. The concept of that technology stack did not exist outside of academia. And so as a scientist would do, Joel Bader, who is another core member of the SC2 sort of leadership, which is to say he was the lab that worked on the design concepts and a lot of the validation at the sequencing level. Joel, and I kind of got together in a hypothesis-driven fashion, said, "Hey, let's point this outward and see if there's any customers." And so incorporated the company, and then it sort of sat idle for a few years while I talked to people and essentially, there was two paths: one, get venture funding, or two, find someone to pay us to do something right out of the gates. And so It turned out to be the second option. And we found a chemical, sort of a top 10 chemical company that was interested in a larger scale yeast-based engineering project. And so with a small amount of money and as luck would have it, the opening of Launch Labs in New York City, which was in the same building that the Buka Lab happened to be located in at the time. We had money, we had a space, and we had a company. And so Neo was sort of born into the physical world, July 1st, 2017. And I remember that because that's Canada Day and I'm Canadian. But anyway, that's what got us off the ground. And from there, it was a lot of inbound associated with Jeff giving talks around the world, and folks sort of coming out of the woodwork and additional projects, as well as SBIR funding, which was tremendously valuable to help fund Neo over those first couple of years.
Amazing.
That's how we got going.
So I wanted to ask about, there must have been a transition for you at some point of, it sounded like at the beginning you were fascinated by the basic science of all of this, right? How do we understand the genome or biology in a fundamental level and how it works? But at some point you start to transition into applications. What are some of the big applications of this? And was there a gradual or a sudden transition where you thought, actually, there's 1,000,001 industrial applications of this core scientific idea that I want to take forward.
Sure. Well, one thing I'll say, I don't actually think I'm a discovery-driven scientist at all. I'm a process-driven scientist. I love thinking about how to do things more efficiently, with fewer reps or more robustly. They work every time, blah, blah, blah. And that's actually 100% how we think about hiring at Neo, because what we do ultimately isn't, I wouldn't say it's repetitive. There's a lot of discovery to be made and how to do things efficiently, and that's kind of what motivates me. But we have to automate and run processes at very high throughput to enable the sort of genome level engineering approaches. What are the applications? Which is a wonderful question. At a high level, I think most of the applications have yet to be identified. We have two great ideas we're working on internally. And I'll talk about those in a second. And the way we're thinking about identifying the whole world of, to be identified opportunities at Neo is really to build an ecosystem of like-minded genome scale thinkers that does not exist today. This is almost a new category on some level. Yes. It kind of depends. Like a whole genome is a lot. If you sort of say, we're going to start the definition of a whole genome at multi-gene pathways, it's much more accessible. But that said, in both of those cases, I think the majority of biotech is at like the base pair gene level change, SynBio being a subcategory. That said, the idea of building an ecosystem is sort of engaging companies across biotech, identifying, that's all sectors, but identifying the immediate near-term needs of those companies and projects that we can work on today. And those are often very bite-sized, but they're valuable because it allows us to develop trust with those new partners and engage in very deep discovery conversations about what it is that's truly challenging to them, and explain the technology, explain the sort of new way of thinking about cell engineering in their domain area, and then sort of go back and forth and identify what those future states could look like for application areas and cell types and domains that I know nothing about.
Yeah.
And so that's been exciting. And all of that is sort of wrapped up into what are the things we can do for you now that leverage our automation and our systematic approach to doing science, onboarding workflows, optimizing them, operating them, tech transferring them back. What are we doing internally? And that's sort of like the second half of the strategy of Neo right now. And that's sort of leading the charge and showing being exemplary about what the technology can do. By building a couple of high-value platform strains. One is probably going to be directed towards gene and cell therapy applications, and the other one with respect to new-to-nature protein biomanufacturing. So we call those NeoVector and NeoYeast.
Good naming.
We obnoxiously put Neo in front of everything now.
Yes, that's right. You got to do it. I'd love to hear about both of those in turn. So what, because I was going to ask about mammalian delivery, and presumably there's a path towards human delivery there. Maybe you could talk a little bit about the gene therapy applications that you see.
Yep. Yeah, that's I think the number one challenge when talking about engineering mammalian cells is delivery. How do you get longer pieces of DNA into cells efficiently? And of course, it's not like it's one cell type, or each species has one cell type. It's many, many, many cell types, and they all have their own, unique challenges associated with response to foreign DNA being stuck into them. So the NeoVector platform is a viral-based delivery platform for high efficiency delivery of up to 150 kb DNAs, which is a lot of payload relative to what is sort of state of the art now. If you say lenti is like top of the line, if you will, you're 15xing that, I guess, or so. So that strain is in development, and I think in 2024, we'll be facing sort of a proof of concept complete if things are going well over the next month or two. And we'll have some exciting decisions to make around how we want to carry that forward.
Yeah, I guess I'm thinking about what can't you deliver with a 10 kb or so lenti that you would want to deliver with 150 kb? Are we talking about tens of genes in a pathway, and you can start booting a cell up with a whole new pathway? What can you deliver that's in that 10 to 150 kb that you couldn't with a lenti?
I think it's pathways, and I think more importantly, it's more natural looking genes with natural regulation, and including introns.
Yeah.
So like something like 90% of human genes are in the length scale of about 100 kb, I should say.
Right.
And so from that point of view, having a packaging system for 150 kb would buy you a whole bunch of additional biology if you care about natural gene regulation.
Yes, that makes sense. And I guess if the yeast, if the SC 2.0 project suggests that introns don't matter, it will probably be on there will still be big fitness effects, right? So they'll matter in some way. What do you think?
Let's just talk about yeast and introns for a second.
Yes.
It's a very different ballgame than certainly human or mammalian cells.
Good.
5% of yeast genes have introns, and those introns are very, very short, like a couple hundred base pairs. It's a very, very compact genome. I think it's apples to oranges in talking about introns in mammalian cells. If anyone is really interested in learning more about this, I think the person to talk to is actually called Yaz Aizawa, and he's in Japan. And he's published some really nice work around the role of introns, at least in a subset of genes, and how that impacts the function of expression, et cetera. And leveraging, he's a product of the Buka Lab as well. Yep. Participated in the SC2 project, and has also sort of gone from yeast to thinking about applying the genome writing style technologies in mammalian cells. And so he's doing some really beautiful work thinking about the role of introns.
Amazing. Yeah. So human introns almost certainly matter a lot. And yes. I guess there's, for treatment of Duchenne muscular dystrophy, there's kind of mini gene deliveries where they chop out pieces of the introns, right? Mm-hmm. But it's likely that for other haploinsufficient genes or other issues that delivering a whole gene with its introns intact would work a lot better. That makes a lot of sense.
Just like tissue specific expression using the 50 kb upstream of even just the coding sequence, that might buy you a lot. It's a gene by gene case basis. But I think that this is a whole world of untapped opportunity with respect to mammalian genome engineering.
Yeah, it's amazing. Okay, so that's NeoVector. On the other side of the equation, you'll have to remind me of the name, but it's about creating new proteins, NeoYeast. Yeah.
So this one is a genome rewritten yeast designed for incorporating non-canonical amino acids or non-standard amino acids into proteins. In this case, you need to eliminate one or more codons globally, and then you can reserve them and repurpose let's just say one codon for use in read through or in programming a non-canonical. So this is a big project. This is not just genome rewriting, it's also protein and RNA engineering. So once you get your rewritten genome, you're still in the position to need to engineer orthogonal translation, which is a synthetase and a tRNA that work together with a non-canonical at the exclusion of all canonical amino acids. Vice versa, your non-canonical cannot work with any of the sort of canonical translation systems. It's a big project. Wow. And really interesting. I think the two big applications we're really interested in leveraging non-canonicals that enable click chemistry. So you can just attach a secondary moiety very efficiently. Lots of applications there. And then protein therapeutics and the use of non-canonicals, in particular with respect to GLP-1, which is the molecules associated with some of the diabetes and obesity indications. We're also interested in that avenue.
Maybe you can say a little bit more about that. So would you use the yeast as a bioreactor to produce GLP-1? Yeah, maybe you can talk a little bit more about that.
This is a journey we're going on currently, contemplating the directing the NeoYeast program towards GLP-1s. The requirement here is incorporating a non-canonical called AIB, whether this is for Ozempic or tirzepatide or others. AIB needs to be incorporated. You can't do that cellularly right now. You need a high efficiency, scalable platform that's engineered to work with AIB. And so that's the sort of, I guess, the problem statement that us, probably others, are going after.
So how are they produced today in a chemistry, like an organic chemistry process or something? This is all news to me. So there's a non-canonical amino acid that's part of these GLP-1s.
That's correct. These are produced at least in part chemically.
Right. And I know there have been scalability issues. I mean, there's been enormous demand and supply issues. Is that part of it, that there's not yet a scalable way to generate these?
The demand is enormous. I mean, I think the expectation is that this sort of class of drugs is going to be the most in demand, highest revenue grossing drug in human history. And so the amount of API required is just going to be enormous. So I can't speak to, what the expectations were by the inventors and the major developers of these drugs, but it's probably far exceeding it. And even their wildest dreams, one would contemplate. And so to that end, identifying scalable solutions is critical.
Yeah, absolutely. Why, you may not know this, I certainly have no idea. Why is there a non-canonical amino acid in these drugs?
I do know. It extends half-life in the body.
Oh, interesting. Do you know which one it is so there's one that breaks down quite quickly and they swap it out and it lasts a lot longer? Is that kind of
That's right. It makes them protease resistant.
Fascinating. This is why I do this podcast. I get to learn so much from people like you.
Well, to be clear, my expertise are in the enabling technology for manufacturing and not in the biology of the drugs themselves.
So in terms of how the construction of entire chromosomes actually happens, maybe you could talk a little bit about that. Are they printed? Are they assembled chemically? How does it work?
Yeah, we design them in silico. Typically, if this is a rewrite of a known existing wild type chromosome, we would start with that wild type sequence and use proprietary is a strong word. Use software, let's say, generically speaking, use software to introduce all of the edits, whether it's an insertion or a deletion, or a sort of a recoding, if you will. That's the third category. Take that sequence, carve it up into pieces, and there's two things to optimize for. The first thing is downstream assemblability, and that depends on how you plan to assemble the longer molecule. And the second one, and this is the sad part, and what I consider to be the dirty little secret of synthetic biology, the synthesizability of that molecule, or that stretch of DNA. Not all pieces of DNA should be considered equal when shipping them out to a synthesis company for synthesis. So that said, we optimize for those two things. The more difficult to synthesize parts will break down into increasingly smaller pieces of DNA, all the way down to oligos. We then source all of that DNA. Once it arrives in-house and it's sort of checked in, we'll start the assembly process. And the reality is that is how chromosomes are built today. And the speed then depends on how efficient we are at each step of construction, which is putting the pieces of DNA together and sequence verifying and make sure we've got what we want, and then how many steps we need to carry out to get to the end product that's going into cells. And so the simple optimizations there really rely on, well, how do we assemble more pieces in one shot? And that's what we've put time into, and we're really good at it, Neo.
So do you need to are there kind of linkers on the ends of the oligos? How does the clicking together of all these tiny pieces actually happen?
Depends on your assembly strategy. This is one of my favorite topics. There's lots of different ways you can assemble DNA. The original approach, I guess, would have been just standard type 2 restriction enzymes. Newer technologies in the last 10 years, Golden Gate, Gibson would be the two favorites. This is not giving anything away because it's published extensively in the literature. Using yeast to assemble DNA is sort of is a very, very reasonable approach, and we rely heavily on that. Sort of by yeast and for yeast is one way to think about it. Yeast is great at assembling its own DNA, and then you can take that same DNA and sort of replace the wild type chromosomes. And so we do a lot of that.
And what are the big barriers to scale right now, both in terms of throughput? And I know it sounds like you're a process person, so I really want to understand how we scale this process, both in terms of throughput, as in going to many more 150 kb deliveries that live and breathe in the cells they go in, and then also in terms of the length. How can we go from 150 to a megabase to when will we be able to sequence whole human chromosome length fragments? And maybe it's a assembly problem at some point, but I'd love to hear about the scaling vectors.
Yeah, you've summarized it very well. Everything you just said. How do you print longer pieces of starting material? So, the steps are: you start with nucleotides, you make oligos, you make double-stranded DNA, and then you make bigger double-stranded DNA. And how do you go from making oligos to making really long double-stranded DNA just by printing it out?
Yeah.
That is a journey a lot of people are going on. We're not actually trying to solve that. We ultimately want to be able to design a very long piece of DNA, order it, and have it arrive the next day. And so there's a lot that underpins that. And we're not necessarily in the game of going from nucleotide. We're not in the game of going from nucleotides to chromosomes. And we have intermediate technologies that allow us to do that. But our big goal is to be able to access those DNAs as fast as possible by whatever means possible to put them into cells. Because what we really want to do is that cell engineering and those applications. Like, that's where we want to live. That whole journey of assembling DNA is one that a lot of people are on right now, and we're happy to participate, and we talk to everybody about that. How do you get it into cells? It depends what cell you're looking at. And there's a lot of people thinking about delivery technologies. Neo is sort of in this position of being, how do I want to say this? We need to have technologies that are useful in the near term.
Yeah.
And we're not, optimizing for 20 years from now. So 150 kb is a very big improvement. It is not the end state that's going to allow this big future of all genomes are designed for biotech purposes, to be very clear.
Yeah. Yes.
What are the sort of near-term, low-hanging fruit applications we can go after with an eye towards the future? This is going to be a stepwise process. There might be a step-full change at some point, and we'll be part of that game, but we will not be the only piece to it.
Yeah, that makes sense. What are the biggest barriers to, if we took the gene therapy application? Is it delivery? Is it something else? What stops us from, there's 7,000 rare diseases, at least 1,000 of them are probably haploinsufficient genes, where if you could figure out how to get the full length gene into someone, it might have a positive impact. What gets us in the way of applying this technology, you think, over the next decade to many thousands of diseases?
Wow. Deep understanding of the biology of the disease and what is actually going to correct it. I don't even know that the Neo technology is the answer. I think it's about a deep understanding of the biology of the disease and having really predictive models, either cells or animals, that impart or actually report out on what will ultimately be the therapeutic.
Right. So if they know what to write to solve the problem, then if you know what to write, you can probably write it and then figure out how to deliver it. But it's the knowing what to write in the first place that is the challenge. Is that right?
I think that's a really big piece of it in the next 5 to 10 years. I think that the 10 to 20 years would be crazy to say longer than that. If the solution requires megabases of DNA and it needs to be delivered systematically, or systemically, excuse me, we need new delivery technologies. And I think it's the piece of how do we know what it is we're trying to solve for, and then how do we get it into the body? And the longer the piece of DNA that's required to solve, the harder the challenge is, I think.
Yeah, that makes sense. Are there any big differences between mammalian and yeast, or a lot of the lessons learned in yeast porting over somewhat nicely to the mammalian genome?
Great question. Well, I mean, the big difference is definitely delivery.
Mm-hmm. Why is that? Why is delivery so different?
A couple of reasons. Yeast is really good at homologous recombination. It's really easy to replace segments of DNA. So if you want to integrate into the existing genome, very easy. Yeast also has very short centromeres, 125 bases. And we understand what all the features are that program a chromosome in yeast, and they're very easy to engineer. So we can build these, we like to call them neochromosomes, shockingly. We can build these things incredibly easily as circles or as linear molecules and have sort of these supernumerary chromosomes programmed and operating alongside the 16 wild-type chromosomes incredibly easily. All of that is harder in mammalian cells. Innate responses are activated, which makes it harder to put DNA into the nucleus. Mammalian cells aren't as good at homologous recombination, so it's harder to have it integrate. Obviously, double-strand break technology has made that more efficient. Finally, chromosome features in mammalian cells are a lot more complicated. Both telomeres and centromeres are far longer and more complicated than in yeast. And so it's really hard to design what we would call a neochromosome that is highly stable and functional and easy to work with, like USB style, like plug and play. Way, way harder in mammalian cells.
Yeah, that makes sense. Last question here, just because we're running out of time. I'm wondering, what application is it that you are really excited about, really believe in, but you just haven't run into that partner who says, yes, I see it the same way. It sounds like gene therapy, there's a big world out there of people who will believe in that. The development of or incorporation of non-canonical amino acids. I also see that. Is there a third bucket where you'd say, I actually really think people should be thinking about X, but I haven't run into somebody who is, and I'm hoping maybe they're listening and we'll reach out to you.
Oh, that's so exciting. That's a hard one to answer. The answer, I should have a better answer than this, I think. What I will say, in the interest of having an answer for you, through conversations in this ecosystem building part of our strategy, we are identifying increasingly more application areas. This is hard to answer because there's confidentiality that I kind of need to consider. I think we're starting to see success in our strategy, which is to say, in this ecosystem building approach, where we're taking on these bite-sized projects and having these deeper discovery conversations, we are beginning to identify these larger scale opportunities and leveling up in these partnerships that we're sort of burgeoning partnerships. That gives me great optimism that this future that I would so like to believe will exist of genome scale engineered cells for biotech. Our hypothesis is more right than wrong right now.
Amazing. Yeah, it seems like you have, like you were saying earlier, it's a new category, and that's both a blessing and a curse because you have so much you can do. You're starting with such a broad playing field. I think the example that people will probably be familiar with are many of the genome sequencing companies like Illumina and PacBio and Oxford Nanopore and all of those that started and had 1,000,001 directions they could go, right? There's so many things you could do with this. So part of the challenge now is, to your point earlier, figuring out what are the things that you can make a difference on in the next couple of years and ladder up to that future, which is, I agree, extremely exciting.
Great. Thank you so much. This was a lot of fun.
My pleasure. Thank you. And thanks everyone for listening. As always, if you have a guest recommendation, any feedback, you can reach us at any time. And like I always say with every single episode, the thing that we'd appreciate most that you do if you like this episode is share it either one-to-one with a friend or a colleague, or put it on social media if you're feeling very brave. That way other people can find us and hear Leslie and her team's amazing work. So thank you again so much. I really appreciate it.